Hey everyone, it's Steven here, and I figured what I would do is to prepare for my microbiology test, I would go through all the different slides we had from the different lessons. So we'll start off with some of the main themes, get into a little bit of the history, and we'll get started with the course. So here we go, main themes of microbiology, chapter 1. And by the way, this is the Cowan chapter 4 textbook. It's called Microbiology, just for reference. So next slide. Let's see. So here we have a picture. This is kind of just to kick us off of a macrophage taking up a lot of bacteria, and we'll learn a little bit about in the course how that works. But basically, the macrophages are like these cells that are lying below your skin surface that are waiting to catch something that's out of order, like a microbe or a bacteria or virus. So we have these in place to sort of keep an eye out underneath the skin and make sure that nothing is going wrong. This is going to a little bit of immunology, but we also have things like dendritic cells and mast cells, which also play a role in that role of being sentinel cells, which we call them sentinel cells, meaning being a sentinel for our body. So what are microbes? Microbes are things that are small that can invade us, essentially. They've had an impact on different areas of the world, such as evolution and biotechnology. This is kind of I was getting off track here. This is more of an outline of what we'll be going over. So I'll just skip this slide then. Microbiology is a study of microbes or organisms too small to be seen with the naked eye. So it makes sense, right? Micro, small, biology, study of life. Microbes are small things. Such things as bacteria, algae, which live in water, protozoa. So protozoa are eukaryotes, so they have a nucleus. So they're similar to us in a way. Helminths, which are like roundworms and flatworms, fungi, so like mushrooms and other things, and viruses, which are pretty commonly known. Just those things that invade cells. Here's some pictures of the different types. We have some bacteria here. We got some of the fungi. These are the little. I could be actually wrong on that. Sorry. We have a paramecium here, which is a type of eukaryote. And I'm not sure what all these are, to be honest. I'm not gonna say what they are. Helminths. So we have these two types of worms that can infect us. We have the flatworms, which are what we refer to as tapeworms. These are the things that people that want to go on like a ghetto type of diet will eat and put in their intestine, and will take up a lot of their food. But do you really want that thing living in you? And roundworms, which I'm told are commonly they infect kids commonly because kids will play in the sandbox or in the soil, and they'll put the fingers in their mouth, and they can get the worms inside their intestine. And from what I'm told, they don't cause a lot of harm, but they're not something you want living inside you. These are some of the different applications of microbiology. So we have a guy inspecting a test tube or a tube at the CDC on a culture of influenza virus there. We have people from the U.S. Public Health Service. They are monitoring and controlling the spread of diseases. You can read all that right there. Immunology, which I might get into a little bit here because the two subjects sort of overlap a little bit. And some more applications, industrial microbiology, because we have things that can infect our food products and beverage products, so we have to have processes in place to make sure that those infections aren't happening in our food. Agricultural microbiology there, and environmental microbiology. I'll let you read that. So the impact of microbes. The prokaryotes were the first organisms to appear on the fossil record. Again, prokaryotes are things that have no nucleus, so we're talking about things like bacteria. Archaea, I believe, are part of that as well. These tend to be things that can live in more extreme environments, so that's why they were the first organisms to appear. Eukaryotes were believed to have evolved from prokaryotes because if you look at a sort of a picture of a eukaryote, what it has a lot of different organelles that have membranes wrapping around them. And the theory on those is that different prokaryotic cells absorbed each other and inherited the functions of those different things that they absorbed, which became the organelles of the eukaryote. So that's just a theory that's going on there. Microbes are ubiquitous. Ubiquitous meaning they're found everywhere, all habitats and environment. But they're absolutely essential to the chain of life as well as the circle of life, if you've ever watched The Lion King. Microbes are also important for photosynthesis as well. So we're commonly told that plants are the main arbiters of photosynthesis, which they are, but they also have there's also bacteria that do photosynthesis as well, things that can make their own energy. Greenhouse gas production, decomposition, so different microbes can break things down. Different fungi are all part of this as well as bacteria. And symbiotic associations with other organisms. So not all infections are bad, right? We have things in our gut that are helping us out. If you've ever had a glass of milk and you know you're lactose intolerant, what will happen is that bacteria in your gut will break down that lactose sugar, and that's what gives you gas. But I guess it's good that you have something breaking it down, right? These are just some pictures. Reproductive structures of spores. Not too sure what to say about these, so I'm not going to speak on them. Biological insecticides. Certain microbes can be useful for infecting insects. So if we are trying to control the insects that are harming our food, then we might use microbes that are pathogenic to them but not to us, such as Bacillus thuringiensis. Don't know if I said that right. Bioremediation. Bacteria can degrade organic matter in sewage. So I believe this is a picture from one of the oil spills in Alaska. So they're using bacteria to help degrade that pollution. Biotechnology is centuries old. There you go. Genetic engineering is where basically you can sort of manipulate bacteria to produce proteins that are good for you. So that's what we refer to when we talk about recombinant technology. Basically taking a gene that encodes one protein and putting it into something else. So in this case, we might use an E. coli bacteria to produce insulin, so that can be helpful for humans. And that's done in the lab in different ways. I forget how it's done exactly, but you can basically get that gene that encodes the protein into the E. coli bacteria to produce those useful proteins for humans. And that's one of the things that we can do with them. Let me take one coffee break real quick. Fermented foods and related products. So we have dairy products like cheeses. Basically bacteria there going on there, bacteria and dairy. By the way, fun fact about cheeses, I would just read that they got, if you have cheese, it's 70% of your calories are from fat. So it's not the best thing for you, but I know it tastes good. So kefir comes from milk. Yogurt, obviously, you can see all these down here. Country cured hams. I never knew this, that those were Cured meant that you're using bacteria to cure it. So that was kind of interesting and a little bit gross, but that's all right. Some more products we have here: cocoa beans, coffee beans, kimchi, so Korean food, and Japanese food, soy sauce. So basically, these are pervading every culture in our world. So no one's really getting away from this stuff. We also produce microbial enzymes commercially, as you can see here, for laundry detergent and cheese production and the lysis of blood clots. You can read all about that there. Those are, microbial enzymes. As I said before, or I kind of alluded to before, human insulin can be made in E. coli and for treating diabetes. Human growth hormone for treating growth effects. Let's see, interleukin 2. That's part of the immune system. So a lot of good stuff that we can make from bacteria. So this was kind of interesting that we have a situation where the FDA is approving a virus as a food additive to help get rid of the bacteria. So, and it's kind of funny because when I saw this, the one thing I thought of was Planet of the Apes, that movie where you have a virus that's causing, it's supposed to help Alzheimer's disease, but it's also making the apes smarter. And so it ends up infecting and killing all the humans and keeping all the apes alive. So I can't help but look at this and not think of that movie. So when disease is caused, when we have a problem in this relationship between bacteria and whatever else we're dealing with, is when the microbe overcomes the host resistance and disease results. Infectious diseases remain one of the most common causes of death worldwide. These are pretty common ones: AIDS, Ebola, and SARS. We also have other infections that are becoming more prevalent because of things like resistance. So bacteria can become resistant over time to certain antibiotics that we're taking. Common one is penicillin, and I know that some bacteria are resistant to this. And one of the things that this is caused by is, I actually just had an infection on my ankle recently, and one of the things the doctor tells you to do is to take all of your antibiotics, because if you don't, then if you take some of the antibiotic and then it goes down and you quit taking it, then that bacteria that's left can develop resistance to your antibiotic and put more resistant strains out into the world and basically harm yourself and everyone else. So measles and pertussis. Pertussis is whooping cough. Fun fact, tussis is a suffix for cough. So if you ever see that word. Historical counts of infectious diseases. So typhoid fever happened in Greece. Don't really know a whole lot about this backstory, so I won't go into it, but it looks like it was pretty devastating. And there's a little map of that area at the time, the Persian Empire and the Kingdom of Macedon. It's interesting how there were so many different places. Bioterrorism, obviously, this is something that we don't really want to happen. Use of viruses, bacteria, fungi, or their toxins for harmful purposes. An example of this was the anthrax scare that happened around 2001 after the September 11th attacks, where different anthrax spores were getting sent out to different congressmen, and that was a problem. So I'm told this is a treatable disease, but it has to be caught early, and so that's one of the things you have to look out for. Early history of microbiology. So we have Leeuwenhoek, who I believe is the father of the microscope. One second. He put together a crude microscope, but he never published his work. And he's also the one we refer to when we talk about the animalcules that he saw under the microscope. We also have Hooke, who coined the term cell and developed the cell theory. There's, I believe, 3 parts to that. You can go look it up. It's not important on the details for this class, but he's the guy that started that in the 1660s. And Antony von Leeuwenhoek in the 1690s was the first person to view microorganisms under a microscope. He made his own little crude fashion one, which I guess I just said that earlier. And I think there's a picture of that later on here. So in cell theory, Robert Hooke observed that thin slices of plant materials with a magnifying glass, he noticed these materials were made of regular organized cubicules that he said looked like gel cells. Interesting, right? And that part of that cell theory, which I mentioned earlier, cells are the fundamental units of life, and they carry out all basic functions of living things. So there you go. If you've ever watched Dragon Ball Z, this will make sense to you because the villain Cell, he was made up of different cells from all the best fighters in the universe, supposedly. And if you were to try and take him out, you'd have to actually destroy him whole, because if he had just one cell remaining, then his whole body could be regenerated from just that one cell, which I know it's far-fetched and it's a show, but it was a kind of interesting biology lesson there. So this is the microscope that Leeuwenhoek used. It had a small specimen holder and lens. It looks kind of like one of those things of jewelry. It's kind of hard to imagine seeing something off of that, but I guess it worked. So before microbiology, what did people from long ago believe was the cause of disease? How did they think a new life came about? Well, let's find out. The golden age of microbiology. So we have Louis Pasteur here. He disproved the spontaneous generation of life. So I think we'll get into this. I can't remember the order of the slides exactly, but I think he proved that basically bacteria doesn't appear out of nowhere, right? So you leave a plate of meat on the table, and what happens after a while, right? You have flies that form around it. You have all kinds of nasty things that happen because something's feeding off of that meat that's less than desirable. But that stuff doesn't come from nowhere, right? So that stuff that's developing on it has to come from something. And so what he did was he did little different experiments where he would put like a glass thing over the meat, and things would develop, but it would take longer and stuff like that. So I think it shows that here. Yeah, here we go. So he basically put a flask of different broth. So it's stuff that bacteria would like. He heated it up, and what would happen is as you can see from the end of this tube here, that bacteria would form at the bottom, but they couldn't get up into the actual broth itself. So the broth wasn't affected. And another word for that is sterile, so it stays sterile. But then one thing he did was he tilted the flask this way so that some of the broth was able to get down through here. And so it interacted with that bacteria that was originally up here. And so what happened is the bacteria would wash back into the flask, and lo and behold, you would see bacteria multiply and grow in that broth. Kind of makes sense, right? So it's feeding off of that broth. But it doesn't come from nowhere. That's important. Pasteur's work led to the development of what is known today as the germ theory of disease, where human disease is caused by infection with microorganisms. Several of Pasteur's contemporaries joined the effort to prove the theory. I'm just reading the slide now. I'm trying to see if there's anything else I need to say on this. Okay. Continuing on with the golden age of microbiology, Lister, who is I believe was the person that Listerine was named after. Industrial microbiology, so Louis Pasteur, we had fermentation, pasteurization, so basically getting rid of the bacteria that's in food, because he linked bacteria to food spoilage. Aseptic techniques, which you learn about in your microbiology lab, basically transferring media from one form to another, or transferring a sample from one media to another, and proper technique is needed to make sure that second form of media is not contaminated by bacteria in the air. So germ theory of disease based on Louis Pasteur's work. So Joseph Lister was a doctor, I believe, and he applied these aseptic techniques for surgery, and he used phenol as a disinfectant. Is there anything else I need to say about Lister other than Listerine? I don't think so. Moving on, germ theory of disease again. Ignaz Semmelweis. He was the guy who basically, in Austria in the 1800s, tested out a hand washing before childbirths, and he noticed that a lot of the deaths from childbirths were going down because of after the washing. And for some reason, what would happen is people thought it was sort of beneath them to wash their hands, or at least physicians did anyway, and so they wouldn't do it. So he kind of had a sad ending. If you want to read about him, he was eventually transferred to an insane asylum, and he was, I think he died by cutting himself on accident during a surgery, and unfortunately, I think that what caused his death was the same disease that he was trying to prevent by hand washing, which really sucks. Robert Koch. I don't know if he's related to the Koch brothers, but he was the first to prove germ theory. He discovered Bacillus anthracis, so that anthrax in the blood of cattle, and developed a series of experiments to prove that a specific microbe causes specific diseases. Florence Nightingale, one of the famous nurses of our time, the founder of modern nursing during the Crimean War. I believe her backstory was that she grew up in the lap of luxury, and she was supposed to marry like a socialite guy, but instead she ended up going to Crimea during the British war effort there, I guess in the 1850s. And she noticed that the conditions for treating soldiers there were deplorable, so she wanted to help out with that. And I'm sorry, I can't tell you more about what she actually did there, but she basically brought a bunch of her cohorts there, and they helped out with the British, because I think now it's coming back to me now from the lesson. So the British Army only had a couple of doctors for lots of people, and so they needed a lot of helps, and so some of the nurses came over. The early nurses came over to help with that. Vaccinations. So here we have Edward Jenner, which I don't know if he's related to Bruce Jenner or not. Caitlyn Jenner. He developed the smallpox vaccine using cowpox blisters. So he would notice that cows were developing these blisters, and he theorized, I believe, that they were the same ones causing smallpox. And so he developed the first vaccine that prevented smallpox in humans. Pasteur, Louis Pasteur again. This guy he's pervasive in this world. Came up with the rabies and cholera vaccines. He's just doing his thing. Elie Metchnikoff was one of the earlier personalities in immunology, so he was a celebrity over there, talking about phagocytosis and defense against disease. And the story with him, I believe, is that he left his lab one day. No, actually, hold on 1 second. So he asked this woman to marry him, or he asked this girl's parents for her hand in marriage, and they approved on the condition that he took in all of their siblings as well, which kind of sucks. But I guess if he really likes her, then maybe the juice is worth the squeeze. But anyway, the other siblings got into his lab one day, and he was working with a bunch of starfish at the time, and what they did was they cut up all the starfish, and he came back and he was kind of pissed off, but he noticed that when he put his starfish under the microscope, he saw that some healing process is taking place by the starfish. And he sort of came up with these ideas of immunology. And I know I sound like a retard right now, but sorry. Golden Age of Microbiology, Paul Ehrlich. Let's see here. Modern chemotherapy. Let's see. He tried to come up with a treatment for syphilis. This was salvarsan. Contained arsenic, but a small amount. It wasn't successful, but it was sort of the first use of a synthetic drug or something that we made ourselves. Let's see, it's Paul Ehrlich. Alexander Fleming, who is pretty well known in this field, discovered the first antibiotic called penicillin, produced naturally by mold, so you could actually make your own penicillin if you wanted to. But it wasn't used clinically until the 1940s. And there's a reason for that, which I can't remember why. It wasn't regulation, which I thought it was, but there's a good reason for the reason it wasn't used until 1940s. Also important to note here, there's a lot of bacteria that are resistant to penicillin now, so it's kind of unfortunate. Taxonomy. Let's see how much more I have left. Okay. Taxonomy is the science of classifying organisms. This is how we get our names from. And we name them for different reasons. We give names to organisms that we do for different reasons, such as common characteristics, and some other reasons that we'll get into in a second here. We call these organisms are placed into taxons. So the guy with the whatchamacallit, the honor of naming these organisms was called Carolus Linnaeus, the father of taxonomy. He came up with the binomial nomenclature, which I believe was the genus and species. So you have the genus of the organism and the species. And let's see here. So scientific nomenclature. The genus is capitalized, and the specific epithet, which I believe used to be called species, now it's called specific epithet, is in lowercase. So as an example, we have the Escherichia coli, or E. coli, which we commonly abbreviate the first letter of that genus. And I guess we italicize both words, yeah. Homo sapiens, that's us, humans. What is in the name? So we name these by shape, nutrient requirement, habitat, sometimes a name after the founder, disease. So here's some examples. Escherichia coli, named after Theodore Esterich. So that's an example of a founder naming the microbe. Hemophilia, Haemophilus ducreae. I think that's hemo is meaning blood and phil meaning love. Named after the Augusto Ducre. Neisseria gonorrhoeae, so gonorrhea, named after the guy. Some more microbes, saccharose is sugar, and myco is mold, and so you can kind of see all these. Staphylococcus aureus is a common one. Staphylos, cluster, coccosus berry. This is kind of an interesting one here. Shigella etuse, named after Kiyoshi Shiga, in the European Theater of Operations of the U.S. Army. So that's the acronym there. European Theater of Operations of the U.S. Army. Interesting. And we got to add the final E to give it a proper Latin ending. So we can also change names, basically because of things like new genetic info. If there's an error in the name that's discovered later on. That was an example of Pastorella pestis. Changes to Yersinia pestis, and it's written like that. Whittaker system of classification is what we used up until recently, where we divide all organisms into 5 kingdoms: the Monera or prokaryotes, the fungi, protista, plants, and animals. So we originally had the 3 domains, or wait, I'm going back on myself. So we had the 6 domains, and we're, I guess, transitioning to a 3-domain system now. I guess things always have to change, right? So we have bacteria, archaea, which still get to keep their own system, but then protista, plants, fungi, and animals are all under eukarya now. Interesting. This is just showing a breakdown of how the different kingdoms are spread out. So we have the different types of trees, the angiosperms and the gymnosperms, the mosses, those are the different types of plants, the fungi, the molds and the yeasts and all that good stuff, algae, archaea. So prokaryotes, so these are things like bacteria. We call this the kingdom Monera. Bacteria and archaea. These are unicellular typically, so they're just one cell. That doesn't mean, though, that they don't interact with each other. That just means that they tend to be made up of only one cell, as opposed to humans, which are made up of tons of different cells. They have different shapes, different morphologies. Is another term for that. They don't have a nucleus, which is important because everything just sort of happens out in the open. DNA, transcription and translation into proteins, it all happens out in the cytoplasm. We have different types of bacteria and archaea going on here. So we have autotrophs and heterotrophs. Autotrophs meaning they make their own energy, and heterotrophs meaning they have to get it from somewhere else. So this is what we're talking about when we're talking about the some bacteria can actually contribute to photosynthesis and release oxygen into the air. Just want to make sure I didn't miss anything with this. Yeah. Okay. Cool. So archaea, these are some examples of archaea. They tend to exist in extreme environments that other things would not be able to live in, which I think is interesting because, we always read about archaea in our biology classes, but they never really explain it to us. It's just kind of something that's out there, and so you can kind of see pictures of it here. So going to eukaryotes, which are some of the organisms that make up us. But first we're going to go into the kingdom Protista, which are protozoa and algae. So we have amoebas under here, parameciums. These are the single-celled ones, I believe. Yeah, so unicellular Protista. Amoebas are interesting because after reading about them, I'm quite confident I'll never swim in a warm water lake again because that's where these tend to live. And if they get up your nose, they can do damage to your brain, which you don't want. And so it's advisable that if you ever go swimming in like a warm standing body of water, that you wear like nose clippings, or you pinch your nose before you jump into the water to make sure that water doesn't rush up into your nostrils, because you don't want that. You don't want those things getting up into your brain. But they do have a true nucleus, unlike bacteria. They can move around, which is good, I guess. They're also made up of autotrophs and heterotrophs, like the bacteria are, so they can either make their own energy or get it from somewhere else. Their typical habitat is either in water or the host. So those amoebas tend to live in water, as well as these other things. So if you take a sample of pond water, look at it under the microscope, and you'll see some of these organisms. Here's some examples: green algae and paramecium. And some more examples. There's an amoeba thing you have to watch out for. A trypanosome. Can't speak too much of that on that one. Here's a pathogenic form of the Protista. So the Mycocutaneous leishmaniasis. And as you can see from the picture here, they have their motile. They have I guess these are flagella on them. More pictures. All right, so we're going to our next kingdom, the kingdom of fungi. So we here have our molds and our mushrooms, stuff like that. So the kingdom fungi, they can either be unicellular, like yeast, or multicellular, like mold. They do have a true nucleus, and they have a cell wall made of chitin. So it's another, just like the bacteria have cell walls, these fungi do. Heterotrophs, so they usually get their energy from somewhere else. They're not typically autotrophs, and they're typically used to decompose things, so that so they're good for that. They usually live in the soil, marine and freshwater, or an animal or plant host. Hopefully not with us, because we don't want them living in us. These cause things like jock itch and ringworm, I believe. Actually, I'll have to check on the ringworm. I know they cause jock itch. I can't remember if they cause ringworm. Classification of eukaryotes. So the Penicillium notatum. These are some pictures of different fungi. The budding yeast cell, that's how they reproduce asexually. Conidia. This is a disease that's caused by fungi. Coccidioidomycosis. Cutaneous blastomycosis, another form of bacteria or fungi, and some different nastier candidiasis, which you'll hear a lot if you get into the medical community. All right, so moving on to plants, things like moss, ferns, conifers, and angiosperms. Checking my notes here to make sure I didn't miss anything. Plants are multicellular. They're autotrophs, and so that means they make their own energy. Moving on to animals, also multicellular. We have to get our energy from somewhere else, because that's why we eat. And other organisms that are under this category are sponges, worms, insects, and other vertebrates. So there you go. And that's the end of chapter 1. So I hope you enjoyed. I'm sorry I'm a little bit monotone, and I did my best to sort of follow my notes and follow the slides from what I remember from class. So if you have any questions, please comment below and let me know. Thanks. Have a good one.